All right, we're going to start off with a quick review of something we spent a lot of time on yesterday, and that was the Calvin cycle and the difference between C3, C4, and plant photosynthesis. And then we're going to hit cell respiration hard here in a second. Remember, the Calvin cycle is the second part of photosynthesis. Where does it happen at? Stroma of the chloroplast. And most plants go straight from the light-dependent part, straight into the Calvin cycle, and we call those plants, what kind of plants? C3s. And they work great as long as what? Not too hot, not too dry, and there's plenty of what in the leaves? Water and carbon dioxide, both. And the way they work it, they take in carbon dioxide 3 molecules at a time. They have this enzyme rubisco, which does carbon fixation, attaches it to a carbon chain called ribulose bisphosphate, 5-carbon chain. And eventually those 6 carbon chains that are made break up into 3 carbon chains we call PGA. We said that's sort of like half of glucose, but it's missing what thing is pretty important? The energy. The energy to charge up this stuff, which eventually will become sugar, comes from here and here. And both of those came from where? The light-dependent part. So part 2 of the Calvin cycle is reduction. And that's using energy from the ATP and the NADPH that was made in part 1 to charge up the sugar. We end up with this stuff, G3P, which we're going to say is the ultimate product of photosynthesis, really. Because that 3-carbon thing can be used to make glucose, which is how we normally think of it. But it can also be used to make starch and cellulose and fructose and any kind of sugar the plant might need. The plant makes 6 of those each turn of the cycle. It only actually uses 1 of them to make sugars, though. The other 5 are used to do phase 3, which is regenerating this stuff, the RUBP, so that the cycle can continue. Now, like I said, this works great as long as there's plenty of water in the ground and plenty of CO2 in the leaves. But when the water runs out, what's the C3 plant forced to do? Close the stomates. What starts to build up in the leaf? Oxygen. And what starts to go down in the leaf? Carbon dioxide. And like we said, this enzyme, rubisco, likes both. It likes carbon dioxide and water. And when there's more oxygen than there is carbon dioxide, instead of attaching CO2 to this stuff, it attaches oxygen. And that problem is called what? Photorespiration. And that doesn't lead to creation of sugar, it leads to the creation of toxic waste product stuff. And if the dryness goes on long enough, what happens to the C3 plant? It either dies or it has to go what? Dormant. It shuts down and goes brown, essentially. Those are C3s. C4s have a little different structure in their leaf. They have thicker cells called bundle sheaths around their veins, and then their mesophils arranged in that around in a circle. C4 plants are plants that are better at living in dry places, like sugarcane, corn, Bermuda grass, sorghum. Their big advantage, their big adaptation, is they have a different enzyme. It's called pep carboxylase. That's out in the middle of the leaf. It has no attraction for what? For oxygen. So they can close their stomates, keep them closed for a long, long time, still fix carbon. But once it gets fixed, they then transport the carbon from out here in this mesophyll area. They transport it in here into these bundle sheath cells, and they let it go again. What enzyme is in here in the bundle sheath cells? Rubisco. But it's in that little small enclosed area. We said it's like putting it in a closet. Even a little CO2 makes what happen? Concentration high. So they have to fix the carbon twice. They fix it out here, where the concentration is low, and then they fix it again here, where the concentration is high. And then they do the Calvin cycle, just like the C3s. But what that allows them to do is close their stomates and keep them closed for a long time. Then open them up, take a deep breath, close them back up. So it's a good adaptation for keeping in what? water. What's that process called that moves water up, through, and out of a plant? Transpiration. So they need to slow that down. Slow it down, keep it in the water. Those are C4s. Now CAMs, the other one that I want to just briefly mention here, are mostly like cactus plants. And like we said a bunch of times, most plants at night, what happens when they do? Most plants. They take a break. They close their stomates, they take it easy. Sort of like we do. But desert plants don't do that. Desert plants, CAM plants, they open their stomates up wide at night. They're breathing fast as they can. And they are using carboxylase, the same enzyme in C4 plants, to fix carbon. And they store the carbon. They store it as an acid over the night. They store as much as they can. As soon as the sun comes up, what happens? Close the stomates because it gets really hot really fast. But this acid that they've been storing up all night breaks back down, releasing a constant supply of what? Carbon dioxide. So that Rubisco, that's involved here too, can continue to do its job all day long. They do the Calvin cycle just like C3s. So these are the best plants at dealing with hot and dry places. And those are CAM plants. Things like the cactus, the pineapple, the orchid. Plants that can handle hot, dry soil. That's all I'm going to say about photosynthesis. Let's switch over and talk cell respiration. I think I'll pick up on this slide, but I'll mention a couple things. What things on Earth do cell respiration? Everything. You got to know the equation for respiration, but the nice thing, it's just what? The reverse of photosynthesis. What are the reactants that something needs to do respiration? Glucose, oxygen, and some ADP and some P. Right? It really needs those things too. And then what are the products? What's the main product? ATP. And what are the waste products per se? Water and carbon dioxide. Because respiration is really just what kind of chemical reaction? It's a combustion. And whenever you burn, whenever you combust the carbon-containing thing, you always give off some form of energy, and you always give off what? Two gases? Carbon dioxide and water vapor, always. So it's a combustion reaction. Remember that the first step in respiration, whether we're talking aerobic or anaerobic, is this one, glycolysis. Where in a cell does it take place? Cytoplasm. Sometimes we also call that what? Cytosol. Either one works. Glycolysis' job is to break up the glucose. So the glucose is a 6-carbon molecule. It's a metabolic pathway. Really, each one of these arrows represents what? What does each arrow represent? A different reaction. What do we know about each reaction? What does it require? And what makes it happen? Different enzymes. So there's lots of enzymes involved. Scientists have found that the reactions and the enzymes that take place on each of these steps are basically the same for what? For everything on Earth. Bacteria, protists, fungus, you. It's essentially the same for everything on Earth. Right in here, actually the first part of photosynthesis, everything on this slide that we're seeing, is sometimes called the energy investment step. This is one of those places where to make money, you gotta what? You gotta spend the money. So what are we actually doing to the glucose in this step right here with one word? Phosphorylating. And then we do it again down here. What's the general name for enzymes that phosphorylate things? Kinases. There's 2 kinases involved here. The kinase that does this step is a really important one. We mentioned it last thing yesterday. What's the name of this enzyme that does this second phosphorylation step? Phosphofructokinase. And it's important for a couple of reasons. The one we want to focus on now, it's got an active site And what will fit into that active site is this stuff, fructose 6-phosphate. And it phosphorylates it. But there's also this other little groove. And what do we call that extra little groove on some enzymes over and over again? Allosteric or feedback site. And what usually binds to the allosteric or the feedback site? The product of the pathway. What's the product of this pathway? ATP. So when a cell has done a lot of respiration, some ATP will bind into this site. What do we know happens? Why is it called allosteric? What does allosteric mean? It means different what? Shape. Just what is that little ATP bound to that site? What does it do to that enzyme? It changes its shape, and therefore, what do we know about that enzyme? It stops working. And if that enzyme doesn't work, what do we know about all the ones that come after? They don't work either. We turn off the whole thing. What do we call that process? Feedback inhibition. We might also call it negative feedback. We might call it allosteric inhibition. It's not permanent. It's just stays that way until what happens? Concentration of ATP drops, and then it'll come off, and things go back up. It's your body being conservative. Your body saving its resources. So what do we know about ATP anyway? You can't store it. So if you make more than you need, you just waste it, right? So you don't want to make too much. You want to make just the right amount. Stop for a minute, use it, then make some more. All right, eventually, all that phosphorylation causes the 6-carbon glucose to break into 2 3-carbon things. Here we call them PGALs. Before though, what else? What do we call this on our glucose slides? Any idea? A lot of books, instead of calling it PGAL, call it G3P. Same thing. G3P. Glyceraldehyde 3-phosphate. Glyceraldehyde 3-phosphate. And what have we said about glyceraldehyde 3-phosphate? It's got what? It's got energy. Lots of energy. In photosynthesis, we were trying to put energy into it. In cell respiration, we're trying to do what? Take it out. One way to take it out is what happens in this step. What's happening to the G3P right here? It's getting oxidized. Electrons are being taken from here and given to that. So what kind of chemical is NAD+? It's a what? Electron carrier, electron acceptor, electron shuttle. It gets NAD+ gets reduced at the same time G3P gets oxidized. So we're essentially moving energy from here to here. Right in here, we break a couple of bonds, and the energy from those bonds is moved into ADP to make ATP. So here we're making a little bit of ATP. Going down, we do that again. Now, is the ATP being made here made by like proton gradients and chemiosmosis? Nope. What do we call the method of making ATP here? It's not chemiosmosis, it's got a different name. It's called substrate level phosphorylation. Taking energy from a substrate, using it to directly phosphorylate ADP. So remember, we put in 2 ATPs at the beginning, we invested 2, but we ended up making what? 4. That's a pretty good investment, right? You doubled your money here. That's always a good investment. You can double your money. We did. And we also got this extra energy product up here, NADH. We end with 2 3-carbon things that are called pyruvates. That's how glycolysis is. So it really made 3 things. Made a couple of 3-carbon pyruvates. We made 4 ATPs, but we used 2, so we usually say we netted 2. We made 2 molecules of NADH. Most of the energy that was in the sugar to begin with, though, is still where when this is over? It's in the pyruvate. So it's step 2's job to get the energy out of here. And what's step 2? If we're talking aerobic, Krebs cycle, aka citric acid cycle. Before we get there, Remember your good old mitochondria. The rest of respiration happens in the mitochondria. Krebs cycle happens in this purple area, the matrix. And the last part, electron transport, happens on the little folds here, and the little folds are called what? Cristae. Also remember, though, between step 1 and step 2, there's step 1B, and it's called the pyruvate conversion. Or sometimes it's called the transition step. I'm going to call it the pyruvate conversion. The Krebs cycle needs to start with a chain of how many carbons? 2. But right now we have a chain of what? 3. So the pyruvate conversion step's job is to oxidize the pyruvate, break off 1 carbon, be left with 2. What's the word for a 2-carbon chain? Acyl. So we're going to take the pyruvate, break 1 carbon off of it, leave 2, and that's called the pyruvate conversion. And we can see that happening here. So the pyruvate, the Krebs cycle starts with an acetyl group, 2 carbons. Let's go to a slightly better diagram here. So this 2-carbon acetyl group goes into the matrix, and sort of like in photosynthesis, first it has to be bound to a carbon chain that's already there. We have 2 carbons, we combine it to a 4-carbon thing called oxaloacetate. In most math books, 2 plus 4 equals 6, right? So we get this 6-carbon thing called citrate, and that's where citric acid cycle comes from. But essentially the main idea of what happens in the Krebs cycle, what happens to these green sugar molecules along the circle? What's happening at almost every step to the sugar? It's getting oxidized. So oxidation happens right here. NAD plus gets reduced to make NADH. So energy was moved from isocitrate into NADH. And as we're oxidizing, bonds are being broken, so what's coming out? What energy, what else is coming out? The sugar's literally falling apart, and it's falling apart into carbon dioxide. So carbon dioxide is being released. Happens again here. Happens again over here. Happens again over here. Remember that this FADH2 is very similar to NADH. It's an electron carrier. It gets reduced at the same time the sugar gets oxidized. So during Krebs cycle, the main thing we make is what? What's the reason for doing the Krebs cycle? What are we trying to make during Krebs cycle? ATP. We'll make just a little bit of ATP though, right? Only 2 per glucose. The main thing that gets made during the Krebs cycle are these guys. NADH, FADH2, NADH. Because in step 3, what are we going to do with all these electron carriers? Yeah, we're going to use the energy from those to make a ton of ATP. That's where most of the ATP is going to get made. The energy to make most of the ATP is going to come from these electron carriers. So we end up with Krebs cycle. Back up the slide here for a second. I've summed together the things that get made between Krebs cycle and the pyruvate conversion together. And this is per glucose. Make 2 ATPs, which is very little. 6 molecules of CO2 are released. That makes sense because we started with C6H12O6, right? So that comes out. We'll make 8 molecules of NADH and 2 of FADH2. And remember, when this step is over, most of the energy that used to be in the sugar is now right in here. And it's the job of step 3 to get the energy out of here and make lots and lots of ATP. Step 3 is electron transport. But I would really rather you think of it as this name, which is the one that people are starting to use more, oxidative what? Phosphorylation. And we'll explain why it's that in a minute. Remember that it happens in the cristae of the mitochondria, and it's essentially about oxidizing the electron carriers, NADH, FADH2, and using that energy to make lots of ATP. I want to go to a blank slide here. draw it up here. All right, so this is supposed to be our mitochondria. What are those red things supposed to be? They're embedded in the membrane. What's embedded in membranes? Proteins. And what are the proteins that are really important in electron transport? Same ones that were important in photosynthesis. Cytochromes. They make up what? What does cytochromes make up? ETC, electron transport chain. That's what they are. They're like the wire that conducts the electricity. Let's go ahead and draw in our other protein here too. It's pretty important. And that protein is what protein? That's ATP synthase. Think about what's going on here. In here in the matrix, during Krebs cycle, the main thing that got made was that guy, NADH. This last step is about oxidizing NADH. So the first cytochrome here grabs onto NADH and oxidizes it. Takes away its electrons, which we'll draw right there. What's left over from NADH after it gets electrons taken away from it? NAD plus and H plus, right? Well, the NAD plus, that's good because it's going to get reused again, right? To do more glycolysis, to do more Krebs cycle. That's a good thing. And then the H plus is contributing to what? Building up the proton gradient, which we're going to need to do to make ATP. The electrons that left, that got taken away from the NADH, move across the ETC. From less electronegative cytochrome all the way down to most electronegative one. And think of it almost like a waterfall. As they move down, what are they releasing? As water falls down the waterfall, what's it losing? As this pin falls, it is losing what? Potential energy. It's losing energy. And in this case, that energy is being used to do what? The energy that it's losing is being used to do what? Pump protons which way? In. Okay, so the protons are getting grabbed from the space here, which, by the way, what's the space in here called? Inner membrane space. Protons are getting pumped from there into the middle of the mitochondria, into the matrix, building up this big H plus gradient. Oh, I said that backwards, completely backwards. Where are the protons getting pumped from? They're getting pumped from the matrix out into the inner membrane space, right? I said it backwards. So proton gradient is actually going to be building up out here. And like we said bunches of times before, as the gradient builds up, it's not stretching a rubber band. Because that gradient is a way to store what? Energy. Because those protons really want to go which way? They want to go in, but they can't, because what's this membrane made out of? Phospholipids, and they don't like charged things, right? So they're stuck. So when a hole opens up in this membrane, what's going to happen? They're going to shoot through, because bigger gradients mean faster what? Diffusion, osmosis. In this case, it's going to be facilitated diffusion, because they're going to go through a protein. And when it opens, these protons shoot in, and this enzyme, ATP synthase, will take their energy, take some ADP, some P, plus the kinetic energy of those moving H pluses, protons, and they will make lots and lots of ATP. Like, was it 34 per glucose that we started with? And then the other thing we need to think about, what's the role of oxygen in this whole deal? Yep. Oxygen will come over to the last cytochrome and take the electrons away from it. Why can it do that? What's oxygen's most famous property, probably? It's really electronegative. So it's more electronegative than the cytochrome. It takes the electrons away, it oxidizes the cytochrome, and when it gets those electrons, what does that oxygen combine up with? hydrogens to make water. And that keeps the cycle going. Now, per NADH, NADH gets oxidized at this first cytochrome. And that causes an H plus to get pumped out right here. It causes another one to get pumped out as it goes through here, and another one as it goes through there. And each proton ends up coming back in here to make one ATP. So one NADH ends up causing the creation of 3 ATPs. Remember that FADH2 is pretty much the same thing, except it gets, instead of getting oxidized at this first one, it gets oxidized at the second one, which means it only causes how many protons to get pumped? 2. So one FADH2 only creates 2 ATPs instead of 3. This is called oxidative phosphorylation because what's getting oxidized? The electron carriers, NADH, FADH2. And what's getting phosphorylated? ADP to make a ton of ATP. That's respiration. That's aerobic respiration. Does that work? All right, let's take our last about 10 minutes here, and let's talk about anaerobic. All right, remember, do you do anaerobic respiration? Yeah, you're not doing much right now, but you would do a lot when what's going on. Yeah, when you're really working out hard, sprinting, lifting really heavy stuff. I'm not talking about a jog. Jogging or why are aerobics, like the exercise, why are aerobics called aerobics? Yeah, in aerobics, you're not going all out, right? You're kind of going half speed. And you can still do aerobic respiration. But sprinting, like running a 100-meter dash, that's anaerobic. You're running so fast that most of your body's still getting oxygen, but what part of your body can't get as much oxygen as it needs? Keep going. The muscles. So your muscles have to have a constant supply of ATP. So if they're not getting enough oxygen, they can't get it from aerobic respiration, so they have to do this in a pinch. Is this very efficient? Nope. In aerobic respiration, if we add up Krebs cycle and glycolysis and electron transport, how many ATPs do we get for every glucose we started with? So somewhere between 36 and 38 per glucose. 36 and 38. And in this process, how many do we get? 2. So it's not efficient. Not efficient at all. But it works in a pinch. It's a way to make a little bit of energy in a pinch to keep your muscles from dying. Can you do this for long term? You know, because when we do it, what's building up? Like acid, and it starts to hurt when we do it, right? So we can't do it for very long, at least not for sustained amounts. There are a few things, though, that only operate through this. Since it's so inefficient, those kinds of things have to be what kinds of organisms? Small and not very active, like bacteria. Some of those are completely anaerobic. The first step is glycolysis. It's exactly like glycolysis in aerobic respiration. It happens in the cytoplasm, cytosol. It makes the same stuff. Two ATPs net, 2 NADHs net, and what else? What's the other product? 2 NADH, 2 ATP, and 2 pyruvic acids. Step 2 happens only if there's not any oxygen around. And step 2 is fermentation. Now, like I stressed to you when we taught this, sometimes some books call this whole process fermentation. Both steps together. So you really got to pay attention to the way they ask questions about it. I'm going to use glycolysis and fermentation as 2 steps. Fermentation, though, its job is essentially to take the pyruvate and the NADH that got made in glycolysis, react those together. Because when aerobic respiration is not happening, what do you start running out of other than oxygen? What do you not have enough of if aerobic respiration isn't happening? NAD+. So the job of fermentation, the real purpose of it, is really to replenish your supply of NAD+. So what it will do is it'll use the pyruvate to do what to NADH? What do we need to do to NADH to make it back into NAD+? Oxidize it. So fermentation is about oxidizing NADH, turning it back into NAD+. If this is happening in say a yeast, they're doing it for this reason, but what's the yeast going to give off as waste? Ethyl alcohol or ethanol, and carbon dioxide, right? CO2. And this is why we use yeast to make bread. The alcohol gives flavoring to the bread. Carbon dioxide does what? It's a rise. No, we don't do that. We have our own version of respiration, or of fermentation, I should say. And in our version, we still have to do it to make this. That's the purpose for doing fermentation. But instead of making the ethanol and CO2, our waste product is what? Lactic acid. Some textbooks, some question writers call it lactate. Don't get those confused. Same thing, lactic acid and lactate. And you'll build that up. Now, once you stop running or stop lifting, what's eventually got to happen to that lactate? You got to break it down and run it through what? What processes has it got to run through? Once you got to run it back through aerobic respiration. So sometimes we say that during anaerobic respiration we build up a debt of what? What kind of debt do we build up? We build up an oxygen debt. Because it's going to take oxygen eventually to run this through aerobic respiration, right? So if you talk about your oxygen debt, all you're talking about is how much oxygen it's going to take to eventually get rid of all that lactate you built up during anaerobic respiration. Oxygen debt. Anybody have questions about any of that stuff? All right, one more thing that I will mention, and then I'll let you go today. We always think about respiration in terms of glucose, starting with glucose. A lot of things that you eat, though, are not glucose, right? Steak, very low glucose in that. Cheeseburger. Cheeseburger is mostly what? Two big macromolecules. Fat and protein, right? Fat and protein. Think about proteins for a minute. It's about the 100th time we've seen this up here. That's what? That's an amino acid. And we know that when you're digesting proteins, we first break them down into amino acids. And then after that, what do we rip off first? The NH2. What's that NH2 group called? The amino group. And that forms what waste product? When that gets ripped off, it combines with the hydrogen to make that. It's ammonia, right? And what organ in our body, what two organs really, have to help us get rid of that? Before the kidneys. The liver is going to take it and convert it to what other less toxic waste product? Urea. And then the kidneys are going to get it out of our bodies for us. But what's left behind is this. That is essentially what? It's two carbons. So it's what? It's an acetyl group. So that's going to skip glycolysis and go straight into what? Krebs cycle. So if we don't start with sugar, we may not start with glycolysis. We may start with Krebs cycle. Fats essentially look like this. I'm not showing it, but what else is attached to each one of these carbons? A bunch of hydrogens. So when you get ready to digest the fat, you're going to break it up into groups of two. And then those groups of two are just going to run straight through what? Krebs cycle. Skip glycolysis. So realize, you always think about it in terms of sugar. It doesn't always work that way because we don't always need sugar. What's the other macromolecule group? Glucose or carbohydrates, fats, proteins, nucleic acids. We use those for energy too. And again, we have to break those down into groups of 2 and run them through crunch time. All right. Good place to stop.